All these open source tools Elizabeth summarized ensure that NeuroLibre has an open climate. And as our developments were ongoing to stitch them together, we had enough resources and opportunity to give NeuroLibre a try. At some point, we couldn't help but cultivate some fields where we can grow living publications. Then we planted seeds of NeuroLibre in open fields, such as eLife and plus computational biology, and even in some other fields surrounded by proprietary fences such as MRM by Wiley or even Elsevier. But in the end, they all grew up nicely and became accessible to everyone. In this open atmosphere, we envision NeuroLibre as an organic partner of OHBM Aperture and looking forward to it coming alive soon. So now, starting with eLife, I will give you examples of how NeuroLibre can invigorate academic publishing. For quite a long time, the microstructural imaging community wanted to have a sense of which MRI measure is the most suitable to quantify myelin, and recently we tried addressing this question with a meta-analysis, which is now published in eLife. Studies included in this meta-analysis differed from each other in various aspects, such as quantified measure, reference methods, or subject type. As you would expect, this non-uniformity doesn't make a qualitative synthesis of the existing literature any better, especially if the data is presented by spreadsheets in several landscape pages presented in a row. But what if there was a better way to distill that information into digestible content, such as an interactive tree map published in NeuroLibre? This figure not only shows how effect size is distributed concerning different grouping conditions, but it is a gateway to every single one of those articles. Then we went ahead and made all the figures in this analysis interactive and created a NeuroLibre book that can integrate our code and the data. Now, when you visit this article on eLife, you will see a link to the NeuroLibre book right in the abstract. As it can bring life to publications, NeuroLibre makes a great tool for interactive tutorials too. A while ago, we were looking for ways to publish a series of interactive tutorials for T1 mapping in QMR Labs blog. After NeuroLibre was born, we converted these multiple interactive blog posts into a single NeuroLibre book. And to do that, the Jupyter book format was really helpful because it gave us an easy way to create a collection of different methods in a well-organized document. Then later on, this NeuroLibre T1 mapping book turned into a book chapter published by Elsevier in the first volume of Quantitative Magnetic Resonance Imaging. And the book itself is, of course, behind the paywall, but it is agreed that the NeuroLibre will be published under the Creative Commons license, and there will be a hyperlink to it in the book chapter. So this way, NeuroLibre was able to go beyond just bringing interactivity to an online code execution to the content, but it naturally moved a significant portion of the end product beyond the proprietary fence and made it available to everyone. And recently, many fields are getting warmed up to the idea of publishing content beyond PDFs. This editorial published in MRM shows that MRI research is one of those fields, and it recognizes NeuroLibre as a platform that can make it happen. So with a small notebook factory team, we are working on converting highlighted MRM publications into NeuroLibre format. We've done a few articles, and we are still working to create more of these. Matthew Baudreau is a NeuroLibre team member. He's working on publishing an editorial in PLOS Computational Biology, and for this editorial, he analyzed the open source landscape of the articles published in this journal. And this analysis revealed that about 40% of the articles matching these keywords of GitHub, open source, shared, and such, they all shared some code, where 75% of them were hosted on GitHub, and about half of them were written in Python. Matthew also analyzed these repositories to find out that about 70% of the shared content was in Jupyter notebooks, and about 12% of all the shared content reproduced their figures. So we can see that PLOS Computational Biology is a decent venue for NeuroLibre. But we didn't stop there by reporting some numbers to show the extent of what is possible we helped create NeuroLibre books for 2+ articles, and they were very well received by the authors. Actually, in one of these papers, we went above and beyond the scope of the article and created interactive figures showing how results would be different if the analysis were run with different parameter configurations. I guess many of us know the story of the Little Prince. He is afraid that some greedy sprouts are going to turn up and take over his little planet, and he works hard every single day to weed them out because he knew if he acted too late, it would be a catastrophe. But he knew that there were good seeds of good plants as well. They would even turn into beautiful flowers he would fall in love with in the future, and maybe that's also why he kept the landscape open. And like the Little Prince, we are putting our best effort to create an ideal environment where good seeds can turn into sprouts of living publications. Because we know that these sprouts will be miraculous when they are fully grown to become NeuroLibre publications. Obviously, NeuroLibre is off to a great start thanks to our amazing sponsors, the Canadian Open Neuroscience Platform, Brain Canada Foundation, Healthy Brains for Healthy Lives, and Quebec Bioimaging Network. We would like to thank Cancer Computing for their generous donation of computational resources and all the contributors from Origami Lab, Neuropoly, and CIMEXP. And finally, thank you so much for listening to.